Everyone will commence very soon. So we would like to have more people on the call. So you can do me a favor by call yet and let them know that we have started. And also you can share the link with the YouTube link with your colleagues, your friends, your family to join this call also. So we'll start we'll start shortly. We'll start in the next probably 5 minutes, yes. We just want to have more people on the call. Thank you. The whole the whole team on behalf of Denmark, on behalf of the director. All right, we are saying congratulations to each and every one of you. Uh, and we indeed see that you are putting much time, much effort, much commitment into doing um, doing well for yourself. And I know that we can all agree. All right, I know we can all agree with me that within this past 3 months, you have even added to your knowledge, and not just added to your knowledge, you've known better, and you've also learned how to do even much better in your field of study. So we congratulate you once again. We hope to see you do even greater things with these skills that you have acquired, with these things that you have learned. All right. So moving forward, I will just give an opening remark, and we will proceed um, to presentations from each group. So for each group, I want us to get ready for our presentation, as this will not take much of our time. Yeah, good evening, Miss. This will not take much of our time. All right. So as we all know that Denmark Institute is given to advancing biotechnology research and education to drive discovery and innovation in genomics, bioinformatics, and artificial intelligence. And we leverage advanced genomics, bioinformatics, and artificial intelligence to develop and propose sustainable solutions to contemporary health challenges. Our research and development efforts focus on cutting edge technology, innovative health solutions, and collaborative research projects. So all of this that we do, it's not just for to gather knowledge, but it's also to be able to apply it and to also improve health. So having said that, I and this is basically because we have other people that are also just planning to apply for the internship, so they can know what exactly Genoma Holdings, Genoma Institute um, is giving to. So once again, I welcome the graduates, and I also welcome those that are joining for the purpose of the orientation. All right, so we will be moving on to the presentation, and the first, all right, okay, before I go on, I would want us to, for those that are just joining, the essence of this presentation is so you can see how much of knowledge the present interns have, and how they've been able to apply to the different projects, and also not just them knowing all of these things, but how it is just beyond giving them, okay, the knowledge, equipping them with what to do, but seeing that, but also seeing that they are able to apply to their project, also seeing that they are able to work with other people from different countries and continents, to be able to come up with something meaningful. So that is the essence of the project, of the presentation of the project, to see how much they've done for themselves, and to also have an idea of what you would also be getting from this internship. So we would be having the first presenter from the comparative genomics group. Yes. So can the presenter for the comparative genomics please come up? And for each group that will be presenting, you all have 5 minutes for your presentation. So please, so as to keep the time and not stay long on this call, can we all stick to the 5 minutes that is given to us? So the presenter for comparative genomics can come up for their presentation.
Hello, good afternoon.
Yes, sir, you're audible. You can go ahead and share your screen.
Okay, so my name is Pascal, and I am representing the comparative um, genomics of pathogenic bacteria group. So I'll be presenting what we have been able to do so far. I wish to use the opportunity to thank Genome Institute for the knowledge impartition. I also wish to thank our instructor together with my team members. All right. So I will go straight to the point. Okay. I hope everyone can see my screen. So our group, the comparative genomics of pathogenic bacteria group, we worked on a research entitled the carbapenemase resistance genes among selected strains of Klebsiella pneumoniae. Carbapenemase, carbapenemase resistance genes among selected strains of Klebsiella pneumoniae. Okay, so I would first want to start by talking of some of the mechanisms of resistance exhibited by bacteria um, against antibiotics. All right, so the first on my list is I have the efflux pump. I normally compare the efflux pump to a child who spits medicine when given medicine. So the bacteria spits out literally the antibiotics when it enters its. So that is one mechanism of resistance exhibited by bacteria. We have the decreased uptake. So both gram-positive and gram-negative bacteria have a peptidoglycan layer. One has inner membrane. And another has both inner and outer membrane. So there are lipopolysaccharides. Some may also form slime layers. So these contribute to the bacteria's ability to resist antibiotics. So this contributes to the second factor of decreased uptake. Okay, so thirdly, we have the inactivation of antibiotics. So bacteria are able to inactivate antibiotics which are ingested. So for instance, many bacteria, for instance, the gram-negative bacteria have um, degrading enzymes in their periplasmic space or around their outer membrane. So these are predominantly beta-lactamases. So the beta-lactam antibiotics, as we all know, consist of the penicillin, the azithromycin, we have cephalosporins, and then carbapenem. All right, so these degrading enzymes called beta-lactamase enzymes. Okay, so there are one family of enzymes, but predominantly the beta-lactamase enzymes are able to degrade or break down these antibiotics, making them not potent. So to kill the organism. All right. So the focus of our research actually sought to look at a particular beta lactamase enzyme called the metallo-beta lactamase, also known as the carbaphenemases. So the carbaphenemases normally attack um, the carbaphenins. So talking of the carbaphenins, we have meropenem, we have imipenem, we have terpenem, and then so these are the vaguely remember. All right. Then we have the alteration of target sites, the alteration of target sites. So as the last mechanism of resistance for antibiotics. Okay, so I believe we all remember something we learned called the lock and key mechanism. So the active site of the active site where the drug has to effectively to work effectively, um, sometimes sometimes it's modified. So making the antibiotic unable to attach in order to act. All right. Um, so that was that I would talk about a few virulence factors seen in Klebsiella pneumoniae. So in Klebsiella pneumoniae, um, the fimbriae, the pilus, the capsule, the lipopolysaccharide, um, the formation of the slime mold. These are some of the things that contribute to the bacteria's ability to cause virulence. All right. So I want to add a few more notes. Okay, so Klebsiella pneumoniae is known to be associated with both community and health care associated infections. And Klebsiella pneumoniae is also known to be one of the escape organisms. Escape organisms, that is one of the WHO priority priority list of pathogens. So the World Health Organization has categorized some pathogens as having developed antimicrobial resistance, needing with a very important and urgent means to um, quickly develop new and potent antimicrobials to solve this particular problem of antimicrobial resistance. Okay, so
All right. Thank you, Mr. Pascal. Please, you can begin to round up your
so
presentation.
escape stands for
Mr. Pascal, please, you can begin to round up.
So,
Can you hear me, Mr. Pascal?
I think in this case, we have Escherichia coli, we have Staphylococcus aureus, we have Klebsiella pneumoniae, Acinetobacter
All right. Thank you very much, Mr. Pascal. I guess the network in the side is not really good. Thank you very much for your presentation and thank you for um, representing the comparative genomics group very well. And your presentation was insightful, and we can see that you have put in much work and effort, not just to and congratulations once again. All right, so before I call on the next presenter, I would just want us to take note of the following instructions and information. Yes, so as regards your certificate, you'll be getting a certificate on Monday. However, we would want to implore each and every one of us to make sure that your payment, you've made complete payment. Please and please, if you've not completed your payment, please do so. If not, you will not be getting your certificate. And also, instructions on how to get a recommendation letter would also be passed across on Monday on individual groups. So please take note, your certificate will be given on Monday, and for the recommendation letter, you would also be given instructions on how to go about getting your recommendation letter on Monday. All of this will be sent to your groups. And like I mentioned earlier, please, as much as possible, as soon as possible, please ensure that you completed your payment. And also for each group that they might not have been done with their work, please and please ensure that you finish up your work on time. The internship, your internship has ended as you are graduating today. However, it will be meaningful that you finish up your work. Also, after today, after your graduation, you'll be given a month extension, and the 1 month extension is for you to finish up your research paper writing, for you to write your research paper. So you can, for each any group that has not finished their research, they have not finished their work, please try to finish up your work within the 1 month and also finish writing your research paper within the space of 1 month. So your 1 month start counting from next week Monday. All right, so I'll be calling on the next presenter from the infectious disease group. So can we have the representative for the infectious diseases group?
Okay, good day everyone. Please confirm if you can hear me.
Yes, you are audible, sir.
Okay. So my name is Ayodele Isaac Adedokun. The group representative for the infectious disease group. I'll be sharing my slide just in a while now. Okay. Sorry, just 1 minute. Okay, you can confirm if you can see my slide.
Yes, sir, we can see your slide.
Okay, is it on presentation mode?
No.
Okay, come, let me just stop sharing and reshare again. Okay, what about now?
It is.
Okay, okay, thank you very much. So, my name is Ayodele Kadedokun, like I earlier said, now presenting on behalf of the infectious disease group. The name here is for me and the staff class rep. So, This 3 months of internship has really been a really engaging one for us. It has been also a moment of discovering more on infectious diseases. And when we talk about infectious disease, what comes to our mind is that we are talking about um, a disease condition that can be transmitted from one person to another, or can be transmitted from one host to another, not just a person, but can be transmitted from a host to another host. Uh, During this period, we're able to see how we can obtain data that are relevant to different infectious agents. Most especially, we focused on bacteria, which are one of the leading cause of the major infectious diseases that we know globally. And for our research, we focused on a very important organism, which is the Mycobacterium tuberculosis. And this Mycobacterium tuberculosis is something that is already known as one of the leading cause of death or mortality globally, particularly in low and middle income countries. So for this 3 month, our internship, we were able to, this is our overview, and through this overview, we'll be going through what we have. So the foundation, which is like our genomic toolkit, during these 3 months, we worked on these five major toolkits. The NCBI, which is the database where we're able to obtain the strains that we worked on for Mycobacterium tuberculosis. So NCBI, we use it to obtain our strains, and through that, we're also able to do one or two from this platform. This database is like an enrichment of information. Then we have the Patrick, which is the BVBRC platform, that's the Bacterial and Viral Bioinformatics Resource Center, which we're able, through this, we're able to do the genomic analysis of our data. Then we use this statistical tool, which is the Tableau, for our data cleaning and every other process, and also visualization, better visualization. Then we use the R Studio to be able to create the charts. So through this chart, we're able to understand what is the relationship between different parameters as relate infectious diseases, from the virulence gene to the antibiotic class to the sample itself, that's the strains, and then the ITO or mega was what we use for our phylogenetics analysis. So Mycobacterium tuberculosis is a gram positive rod. Please, if my slide is not moving, you can just let me know. And also, if you can't hear me, you can also let me know. Hello?
Yes, yes,
I can hear you.
You can.
Okay.
Yes, sir.
Okay, so I said Mycobacterium tuberculosis, when we hear about it, is the gram positive rod-shaped bacteria that is from the family of Lactobacillus and the phylum Acinetobacter. And this bacteria is something that can be spread through the air when someone that is having an active disease, cough, sneeze, or talk. So this is something that can be transmitted through air droplets, through aerosols and everything. Then this bacteria is also an opportunistic and intracellular pathogen, meaning that it thrives in the presence of immunosuppression, or when someone is immunocompromised, you see this bacteria in that particular disease condition. That's why it's mostly related with HIV, malaria, and other disease condition that suppresses the immune system. Then it's also intracellular, that means it's very hard to kill. And this the condition can either be preliminary or extra preliminary. Preliminary, that means is affecting the other organs. And it's also resistant to multiple antibiotics, including the second line injectable drugs. Then the significant public health concern of TB cannot be overemphasized. Then after we're able to choose Mycobacterium tuberculosis as our strain of choice for our research, we started with data retrieval. And the essence of this data retrieval is for us to be able to get information on the different strains of Mycobacterium that is implicated around the world. We didn't just focus on one particular continent. We wanted to take it from a diverse view. What is the implication of Mycobacterium tuberculosis across the different continents in the world? And using our NCBI pathogen detection browser, we're able to do what we call data query. And through this data query, we're able to query our data or our strain based on the region, based on the isolation source, based on the strain, and also we also did that based on the SRR number, which is part of the SR, that's the sequence read archive accession number. Then we retrieve 10 strings across 5 continents. So across the 5 continents, we're able to retrieve 10 strings of clinical importance, and that's what we're able to attain from our filter. So from this image I have here, just like a pictorial image of what we're able to do on the SCBI pathogen detection platform for figure 2, while figure 3 is showing what we did on our metadata. So after we're able to do the data query, we're now able to do data integration, where we focused on the bio project, the bio sample, the SRA number, the genome length. So we want to know what is the length of the genome of that particular strain. Where is this strain being isolated from? Is it from sputum? Is it from blood? Is it from environmental source? And every other. Then the sequencing platform, because these strains now that have been sequenced is more of the whole genome sequence. You want to know what is the whole genome of this infectious agent. So the sequencing platform for some is Illumina, for some is the Oxford Nanopore, and through that we're able to understand what is the complete genome sequence of this bacteria to inform our results. Then this slide talks about how we're able to get our results. So like I said, we try to take it from a divergent view, with so as to understand what is the public health relevance and diversity of Mycobacterium tuberculosis. And we took from South Korea, from Spain, from China, Egypt, Morocco, USA, Canada, and Belgium. And these are like the countries with increased public health relevance and diversity as of recent data. Then the next thing we now did is to do genome assembly. So we already have our raw data. We now want to assemble this data. And to perform genome assembly, we use the pipeline known as the BVBRC. This BVBRC is an enriched platform which allowed us to have access to genome annotation, comprehensive genome analysis, even for metagenomic data. That means if we're going to check the relation to the environment. And for the genome assembly, what we used was our SRR number for the genome assembly, which we're also able to obtain from the NCBI platform. So the NCBI platform gave us that access to the SRR number. And after that, we pasted it on the BVBRC. And that's for the image, you see where they wrote SRA run assertion. So you put it there, and after that, you validate. You need to validate your SRR number. For some of the SRR numbers, they are not on the BVBRC pipeline, so you need to validate it. So if it's not there, then you need to work on the paired read library and the single read library. So for this, it means that for paired read library, there's a forward and a reverse read. Right? For single read library, it's just a continuous read. Then you also need to do what we call more of you're doing quality control. So for that's where you go to the parameters and you do some sort of quality control before you do your assembly. That means you want to trim out the reads. There are some reads that is not really necessary in your assembling data. So you need to clean it out. Then you now carry out the genome assembly and you download the file. So after your genome assembly, what you obtain is your config file, which is in FASTA format. So we have different formats. We have the FASTA format, we have the FASTQ format. These are different formats where you can obtain your data, which contains now the whole genome, the nucleotide sequence of this particular strain. Then the next thing we did now is to do now a more comprehensive form of analysis, which is the comprehensive genome analysis, which covers our functional annotation and our feature extraction. And we also did this using the Patrick or the BVBRC platform. And for this, what we did is also to now check for each strain, because from this now we'll be able to understand this strain belongs to this family, belongs to this class, belongs to this order, what is their genome length, what is their contig L50 and their contig N50 length, what is the GC content is very, very important. That's the guanine to cytosine linkage. What is the content? So for a good analysis, your GC content should be above 55%. So for this, based on the figure we have here, our gene content was around 65.4, which shows that we're working with strains that will give us a very comprehensive image of antimicrobial resistance. We also check the tRNA and the rRNA, which is transfer ribosomal RNA, and also give us a better information on the hypothetical proteins. So these are more of the proteins that play a major role in resistance pattern of Mycobacterium tuberculosis. Then after that, we have to do that organization. And for this, we're able to understand what is the plasmid number, because for these bacteria, for bacteria in general, they see that we have a plasmid DNA or a chromosomal DNA. So for this, we're able to understand based on the horizontal gene transfer, is it having plasmid or not? What is the RNA genes that is contained in this bacteria? And every other thing that we need to know. Also, the genome size, we're able to know about that. The open reading frame, that's like the continuity of that bacteria, we're able to get more information on that. Then after we're able to do this, our data integration, our data retrieval, we're now able to do our processing of this virulence gene. Because for each strain, we're able to obtain genes that cause this resistance, because this essence of this internship was to understand what are the genes or what are the factors that could lead to antimicrobial resistance or antibiotic resistance. So to do that, to categorize them, we're able to use Tableau, which is a statistical tool, and through that we're able to categorize our obtained data. We're able to do gene against the sample strain, classification against the classification against sample strain. We're able to do the antibiotic class against the sample strain. We're able to do some other things too against the sample strain, and through that we're able to now see more information. So after we finish our data categorization with Tableau, we went ahead to do visualization using an heat map. So from this image you are seeing here, this is an heat map, which is the essence of heat map is we are trying to use color coding to show the relationship, the linear relationship between two variables. So for this now, what we did is that we were trying to check what is the real linear relationship between our gene, that's the virulence gene, against the antibiotic class. From here, you see we have aminoglycosides, we have beta-lactam, we have the ethambutol. These are all drugs that are being used against TB infection. And for some, we had to do them multi-class. So this multi-class means that this is not just only one drug. There are multiple drugs against that particular infection, that's that particular strain of gene. And this green one and the red is just trying to tell you based on the grading. So for the one that is red, it means that what there's high resistance to the more number of genes. Yeah. So let's say, for example, now we have the peptide antibiotics, which is indicating in form of red here. It shows that most of the genes, that means the antibiotics now is not really having, is not functional, is not sensitive to most of these genes. So when the difference of this gene in that particular strain, you're giving a patient these antibiotics is not going to do anything. It's just going to be their resistance. Then we also did the strain against the gene. So we're able to also understand what is the strain there. And for we observed that among all the genes, the GDPD was like one of the strains that was more common among all our strains. So that's like this, the gene that had more linear correlation with the strains that we use across all the old countries that we took around the world. Then after that, we're also able to do what we call functional genome characterization. So this is more of you trying to check the roles of this genome in metabolism, in virulence, and also stress response. And for that, there are some, let me call the components that influence these processes from metabolism to virulence to stress response. And those are things like the amino acid and the derivative, the cofactors, which these bacteria require to be able to survive for growth, for resistance, for bypassing immune system. Then we have stress response, defense, and virulence. This is when you are talking about maybe there's more of there are some things that the bacteria does that is going to stress the cell, and also from there they can now begin to infect other cells. Then membrane transport, this is like being able to carry nutrients from one particular cell to another particular cell. This is what you usually say, like for mostly bacteria and MTB, they require like iron. The element iron is being transferred in a place where there's iron limited condition. The carbohydrates, which is like the backbone of the mycobacterium uh, tuberculosis cell wall. That's the glycogen and every the mycolic acid and everything. They're like the backbone of. So it gives the mycobacterium tuberculosis more rigidity. So for you to even kill it, you know that the cell wall alone is rigid. So drugs find it very difficult to penetrate the cell wall. So this is what contributes the to that complex structure of mycobacterium tuberculosis. Then after that, we're not able to understand what is the correlation. Then we are trying to understand how closely are these strains to themselves. We worked on 10 strains. So we want to understand what is the relationship between all the strains across the continents. Through that, we're able to achieve that using our phylogenetic tree. And for this is the one of ITOL. We also have Mega. So we can also use Mega to also generate this relationship between the strains. So what is the relationship across the different locations from Egypt to South Korea to USA to Canada? We need to understand this relationship. And through phylogenetics, we're able to have a better understanding of
Isaac, your voice is not clear.
Hello, we can't hear you.
can you hear me now?
Yes.
Yes.
Okay. So I don't know why I stopped when my voice stopped, it went off. So I was talking about phylogenetics, that this is like us trying to understand relationship.
Yes, you've talked about that. We heard all about phylogenesis.
Okay, so I think someone is muting everybody. That's why I was also muted. Okay, so I said my reflection on my learning journey has been a great experience. And through that, I've been able to gain an hands-on experience with platforms like BVBRC, NCBI, Mega. I've been able to learn how to retrieve, assemble, and annotate genomic data. I've been able to gain skills in using Tableau and R, which not just only based on what we learned, I was also able to improve myself to also do some other chart analysis like histogram on R. And I'm also able to understand how genomics supports public health surveillance. So my passion for infectious disease research began during my research internship at Federal Medical Center of Kebbi, Nigeria, where I worked on inflammation-resistant mycobacterium tuberculosis. And through that, I saw how this infection is affecting the vulnerable population. And that's what influenced me to focus on infectious disease and cause. And this internship has really been a step in that journey, given the tools I've been able to work on, not just as a scientist, but as someone that is interested in translating lab work into tangible patient benefits through my research. So through this is a summary of both of the research we have done and our experience during this 3 months as a group. Thank you for listening. Okay, so I'll stop sharing my slide. I don't know if the host is around. Hello, please confirm if you can hear me.
Yes, I can hear you. I
Yes,
can
I
hear
can
you.
hear you.
Okay. Thank you very much. I am through with my presentation.
Hello, everybody. Good day. Can you all hear me, please? Can you hear me?
Yes, yes.
All right. I think the anchor had issues with network, so she will join us very shortly. But also, I will be going straight to the orientation for the new cohort. Once again, I want to welcome you to and congratulate you for successful completion of your internship program here at Genomark, and also welcome those new interns that will be joining us in the next cohort. And the presentation has been so wonderful. Actually, I enjoy listening to it, and that shows the level of hard work that you have put into not just the presentation, but the level of dedication that you have put into your internship here at Genomark. We are really proud of you, and we are glad to be part of your success story in this journey of bioinformatics. I can tell you that this is just a starting point. Along the way, uh, you may experience some challenges, but you shouldn't give up. That is why we are charging the course of research in, in bioinformatics. So I would like to go straight to my presentation and orientation for the new cohort that is coming. So please bear with me as I share my screen. Can you all hear me again?
Yes.
So I would like to start by introducing to us Genomark and what we do and the reason for this internship program. So Genomark started with embodying a passion to see that bioinformatics skill is not just limited to some specific field. Please, if you can hear me, I want you to say yes. Can we hear me? Can we hear me, please?
Yes, yes.
All right, so I will continue. So I was saying that Genomark started with embodying the passion to make sure that bioinformatics skill is not limited to specific field. Even before the bioinformatics as a whole became known in Nigeria and Africa as a whole, countries abroad, countries outside Nigeria have been offering courses in bioinformatics and all those, even to the master's level. So Gentleman came as the channel to bridge the gap between between to bridge the skill gap between those who have the skill and those who do not have the skill. So that is one of the reasons why this internship program came. And the main reason behind the internship program is for empowerment, is social impact, to make sure that we can have as many people that knows about bioinformatics, to make sure that we have as many people that knows about bioinformatics. And so far, Genomark have been able to empower more than 100,000 people in the field of bioinformatics all across the globe. That is as far as we have reached in making sure that bioinformatics and genome courses is made available at a very cheap rate to everyone that is interested. And along the way, we are really happy with the result that we are getting. not just in Nigeria, not just in Africa, but all over the world. So this course is beginner friendly. As you can see, those who just presented how they were taking from fundamentals of bioinformatics to a place that they can even have a research paper on what they have studied in the 3 month internship program. So it is a beginner friendly, no matter the level of your level of career, either you have heard about mathematics before, or maybe it is a friend that told you, or you just researched online, or you were reading through some papers and you discover that genomics and bioinformatics is very important in the 21st century. We at Genomark, we are here to make sure that the necessary skill that you need to kickstart your career and to build on the career is made available to you at affordable price. Actually, that is the mind for our internship program. And we can also tell you that in Africa and Nigeria, we are one of the leading companies making sure that these skills is made available. So it is the bioinformatics, this virtual internship program is 100% virtual. And what I mean by 100% virtual is that you have access to pre-recorded classes that you can watch, that you can go through at your own space the um, within the time allocated for the internship. So it is career transforming. Are we together? Are we together, please? Are we together?
Yes, we are with you. We are with you, sir.
All right, thank you.
Hello, we are there.
So why do we need? Why do you what? Why do you join this internship program? One, this internship program is designed to skyrocket your career. Very recently, I was filling an application, and the application was strictly for those who have skills in the area of bioinformatics. So there are some opportunities that is coming out now because we all know that the world is going to the area of precision medicine, and bioinformatics is one key aspect of precision medicine. So we have some opportunity that those who do not have the skill in bioinformatics and genomics will not be able to apply for. So it is designed to skyrocket your career. It is designed to position you ahead for the opportunity that will be coming and that is actually made for some specific field. So either you are starting out, maybe you don't know anything about mathematics, you have never done anything related to it, or you are trying to begin on it. So we are here to make sure that you have the tools, the mentorship, and the global exposure that you need to stand out anywhere in the world. So anyone that applied for our internship program, they can all testify to you that they are getting more than what they are paid for. That what we are offering or what we offer them was way beyond what they paid for. So this course is almost like free of charge, and the money that you require to pay, it's for commitment fee, because we know that we have, due to our experience, we have realized that sometimes when there is not a price tag to what is being offered, people tend to take it for granted. So you are getting something worth $3,500, and if you convert that to naira or to your currency, if you are not in Nigeria or everywhere you are, you can know what that was. So, and you will gain expert mentorship. Like when you are going through this internship program, the classes are designed to be virtual, right? But yet we are not leaving you to just watch the courses alone. Every week there will be a live session with a mentor that will be assigned to each program to each course. So it is that mentor that will make sure that you are well guided in the course of your internship. Maybe you have questions in some specific areas that you don't understand. There will be the mentor is there to guide you and to explain the courses to you more. So we don't just leave the course to you. We make sure that we see you through your internship program. It is also um, we also have a research platform which is called the OMIS board. And at this junction, I would like you to show. I would like to show you the OMIS board, and so that you can see. Hello. So that you can see some of our courses and what we offer. So this is the OMIS board. So you can just go online and search for www.omisboard.com. So when you come here, you have this interface. So anyone that for any course that you register for, maybe you register for cancer genomics, all your courses is already in cancer genomics. If it is practicing disease, the courses are already there. You'll be given access and so forth and so on. So if it is prostate cancer, human microbiome, epidemiology, infectious disease, virulence, mobile gene element, comparative, etc. No matter the course that you apply for, there is already a structured course designed for that course. So that is sorry. So you have access to a 3-month license to the OMIS board for seamless research tools and submission. And on each course, you have access to some other materials like textbooks that you can download and read at your own space. You also have access to some software application like the Genius software. If you want to download, or the Genius software is a paid software, and if you want to pay for it on yourself, you may have to pay like $500. So, but we make it available to you free of charge. So the worth of that OMIS board is already up to $1,000, and we are not charging up to that. So you have skill advancements, and you have explained bioinformatic tools that I've said. You have various resources that you can access, and very importantly, just yesterday, one of our interns reached out to me that one of the reasons why she applied for our course is because of the publication. And so it is not just that you are going to the class, but it is a practical class. It is an hands-on class where you have to engage in practical projects, and at the end, those projects forms your research project and publication. So you also have access to publish paper with us. And then, as I've said, it is very beginner friendly and it is career purpose. Some other bonuses that you will stand to get is the recommendation letter, and our recommendation letter is very is recognized worldwide. At the end of this program, you also have a certificate and we have a structural learning. I have shown you some of the programs that you can do with us. We have the plant genomics, the viral genomics, we have the cancer genomics, infectious disease, human microbiome, and etc. And for this new cohort, we will be starting by Wednesday, 8th of October, which is next week. So everybody that we want to be part of this program must make sure that his or her commitment fee, which is $30, is paid before then to secure that slot, because we have limited space. So anyone interested can pay before that. I have sent the link. I have pinned the link on the group, so you can just go to the chat on the group, and you will see that I pinned 2 messages there. You can use those links to join our WhatsApp group, and another link you can use it to access our website. So the program begins this Wednesday, the 15th of October, and it will end on the 14th of October. And you can see the outline of the program. I will also want to know that the commitment fee is a non-refundable fee, and it can be paid in installments. Um, so we have various internship policy, and we align this policy to anyone that is interested and applied for our course. So thank you so much for having me. I must confess again, I really enjoyed our presentation. And if there is any question, I would like to take those briefly before I hand it over to the anchor to continue. So if there is any question, you can ask your question now before the anchor takes over. Is there any question? So thank you so much for having me. I would like to call on the epidemiology group to present. So go for their presentation. The epidemiology, can you please, who is the representative for epidemiology? If you are ready, please, you can share your screen and start your presentation. Um, epidemiology, please go ahead
Okay,
with your presentation.
I'm trying to share my screen. Sorry. Just a minute, please.
All right.
Oh my God.
Before epidemiology comes up. So please, all right, the question says, can we do more than one course? Yes, you can register more than a course, but it will be each course is carries a weight of $30 commitment fee, but there is room for more than one course. But I will not advise more than two so that you can get the best out of your internship program. Thank you so much.
All right.
Okay. Sorry, I have a question.
Okay.
Yes. For the omics board, are we going to still be having access to it after the internship? Because we still need to refer back to it for our research writing. So I want to know.
All right, sir. The Homies Board access 3 months, and it covers only the duration of your internship program. Ideally, we don't give access to that we deny access after the three months. But in some exceptional cases, we do extend the access to Homies Board maybe for 1 or 2 weeks, or sometimes 1 month. Though ideally, our policy is that after 3 months, we should not have access to it. But yet, we still consider it in some cases so that you can round up with your project courses.
Okay. Also, for our own court now that is just graduating, are we going to be having that grace because we still because of the timeline and delays and everything, and the way we're unable to carry ourselves along. Because for my own group, I can say we're just like five that did everything. So the workload was much on us. We were not able to do it among other group members. So can we get that extension?
Yes, you can get a senior, but I would like you to, you know, you have coordinators on your group. So just reach out to the coordinators, or
Okay.
better still, you can just post it on your group to request for extension of access to this board. So it will be taken up from there.
Okay, no problem. I'll do that on the group. Thank you.
Thank you. Epidemiology, please
Okay.
start your presentation. Thank you.
I don't know if you can see my screen. Good evening, everyone.
Yes, we can see your screen, sir.
Okay, my name is Promise, and we worked on a project on uncovering the resistome, a metagenomic is one of the major global health threats. It has already been projected to cause up to 10 million deaths every year by 2050. Now, hospitals are major hotspots because both antibiotics and resistant bacteria from patients often end up in hospital wastewater. So now this wastewater then mixes with the environment, creating a pathway for resistant genes to spread into the wider community. Well, while several countries in Europe and Asia have studied hospital wastewater, in Africa, there are just very few genomic level studies. So our study decided to provide one of the first resistant-wide analysis of hospital wastewater in Africa. We used metagenomic sequencing data from Cameroon, Uganda, and South Africa. Well, we had just three objectives. We first wanted to describe the general genomic features of hospital wastewater metagenomics. Then also we wanted to identify the functional subsystems that support microbial survival and adaptation. And lastly, we decided to perform a resistance profile analysis, linking resistance genes to antibiotic classes, resistance mechanism, and the bacteria families carrying them. We retrieved 11 wastewater metagenomic datasets from the NCBI public database, 5 from Cameroon, 5 from Uganda, and 1 from South Africa. Now, these datasets, they were sequenced on Illumina platforms. We performed quality control, genome assembly, and annotation using the BVBRS pipeline. And for functional and resistant profiling, we use the softwares here, that's the RAST and the Patrick AMR pipeline to identify genes, antibiotic classes, and resistant mechanisms and their bacterial sources. Now, for the results, we got like 3 results specifically. One for the genomic features. The assembled genomes range from about 15 million bases in Uganda to 46. Then GC content varied widely from 42% to 62%, showing different microbial community structures. Some samples contained more than 60,000 predicted genes. However, less than half of these were annotated, meaning that large proportion were classified as hypothetical proteins, which actually highlights that there is huge unexplored diversity of bacteria in African hospital wastewater. Now, for the functional subsystems, subsystem analysis revealed consistent enrichment in amino acid metabolism, protein synthesis, stress response, and virulence genes. Stress and virulence genes were particularly high in samples from Cameroon and Uganda, suggesting selective pressure in wastewater environments. Now, also we got results from the resistant profiles. Firstly, for the antibiotic classes, we got that the beta-lactams were the most abundant across all countries. Resistant mechanisms, the main strategies included target alteration, enzymatic inactivation, and efflux pumps, Efflux pumps were especially enriched in Guardian samples. Now, for the resistant genes, we identified clinically relevant genes, including BLA and some multi-drug resistance clusters were especially evident in Uganda and South Africa. Gene antibiotic mapping. BLA genes mapped to beta-lactams. And also, we also got that resistant genes mainly originated from some diseases which were bacterial families well known from hospital associated infections. Now, in the conclusions, we have to conclude from the results that we got that African hospital wastewater harbors diverse resistant genes, including resistance to last-line antibiotics such as carbapenems and clibopeptides. We presented one of the first system-level resistant profiles in Africa, linking genes to antibiotic classes, mechanisms, and bacterial hosts. These findings highlight wastewater was as a critical surveillance target for antimicrobial control. Our results emphasize the urgent need for environmental monitoring to complement clinical antimicrobial surveillance and to inform policy interventions. I would like to just acknowledge my colleagues and our instructor for his guidance and also the public data set that we used. Thank you very much.
Thank you very much for your presentation, Mr. Promise. And thank you very much for staying with us till this moment and speaking to each and every one of us on the call and those also joining us on YouTube. Deborah Gift, Temilade, Olarimide, Abdulganiu Nofisat, Shalom, Ogunshola, Tosin, Isi, Destiny. Thank you very much for still staying with us till this time. Thank you to each and every presenter and each and every group that has come up for their presentation. We once again celebrate you, we once again rejoice with you, and we can see indeed, not even by we just saying, we can see indeed that you have learned so much and you are even much more better than what how you started. So we really celebrate you. We really celebrate you. All right, so I would take the announcement once again. I made some announcement earlier for those that are not were not on the call. We okay concerning the certificate, I mentioned that you'll be getting your certificate next week Monday. You are expected to drop your receipt on the group, and you would your receipt for your full payment, whether you made the payment twice, made it thrice, just make sure you send your all your receipts to the group, and you will be giving your certificate on Monday. And for the recommendation letter, also you would also get instructions on how to get that on Monday. All of that will be sent to your individual groups. And as regards, I think one person, Mr. Yodele, raised a concern about the about access to make God after. Like my colleague has said, you can do all that you have said, and we will also get back to you on how long we would be extending, if we'll be extending access to Domix board. I think all has been said, everything has been addressed concerning, and for those that have not finished their work, for each group that has not finished their work, cancer genomics, plant genomics, and even the groups that might have presented, this would you have a 1 month extension, and the 1 month extension is solely for your research paper writing. And if you have not finished your work, you can do well to finish it up within that 1 month. And your 1 month starts counting from Monday, 6th of October. So we want you to all answer. I want everyone to work much more for each team that might not have been doing well together. We would implore you all that you work together much more to us finish your project and to have um, meaningful results all your hard work for the past 3 months. All right, so on that note, we will be ending this meeting. Once again, congratulations. We celebrate you and we hope to see you do great and better things even from here. Do well to enjoy the rest of your day. Thank
तुम सब की माँ की चूत भोसड़ी वालों हजारे ये क्लास को लेने अंग्रेज है ना hello motherfucking fellas fuck you all whoever talks now is gay hey, yo,